Amazing. So I'll give a very short intro. Today we have the pleasure of having Dr. Romain Lopez here with us. He's an assistant professor of computer science and biology at NYU, and we've been following his amazing work for years now. To give a little bit about his background, he received his Master of Science in Applied Math from the Four Polytechnique, his PhD in Electrical Engineering Computer Science in UC Berkeley, advised by Michael Jordan and Nir Youssef. And in his PhD, he already did really influential work. I think everyone here knows about SCBI or SCBI tools, and really the flagship paper on deep generative modeling to being a magnet for so many talented students. Some of them we were fortunate to come to our lab, including Justin, who's here. Shield, who graduated last year, that we were fortunate that they were trained by Romain. So in his postdoc, working with Aviv Regev and Jonathan Pritchard at Genentech and Stanford, he did even more incredible work bridging deep generative models with causal discovery. And we're probably hearing some of that work today, and also doing, not just modeling cellular responsive, but also dynamics with flow matching. So really exciting work, and really looking forward to that. So, thank you very much.
So I'm very happy to be here and to be visiting Colombia. I just moved to New York in April, so this is my first time coming this far uptown, and I hope to come back many times and to start a great intellectual relationship with here. And I think there is a lot of momentum that we can get started on collaborations across Manhattan. So today I will not talk about my work in my lab because the lab is a bit too young for that, but I will talk about some work from my postdoc that is still unpublished, but we're always saying published soon, so it's getting closer now, and I'm very happy to be presenting it for the first time. So this is actually the first work I wanted to do. The first project I wanted to work on when I started my postdoc. I was really excited about imaging and sequencing cells in the context of perturbations, and trying from a purely technological standpoint, understanding what do we gain from integrating those technologies and what new biology we can learn from it. So this is joint work with my dear friend and colleague Taka Kudo, who's a postdoc at Genentech. And we've been really very happy to get the help from Katie and Aftar, who were both experts in optical prescreens and perturb-seq. And this was joint work, of course, with my co-mentors, Jonathan Pritchard and Aviv Regev. So the use case we're going to study today is macrophages. So usually people think about macrophages because of their phagocytic activity. So macrophages can digest and eat antigens. Here is an example, and it's usually what we think about. Macrophages, for example, who are able to eat bacteria and degrade, and it's an important part of the innate immune system. We can see that in the tens of seconds, we already have this really nice activity here. But that's not the only thing that they do. Okay, so macrophages are also have important roles in not only inflammation and phagocytosis, but also in regulation of the immune system. And for example, when those functions are dysregulated, it usually entails disease. So for example, overinflammation can appear in type 2 diabetes, as well as inflammatory bowel disease and rheumatoid arthritis and others. And when you have a loss of a balance that is disrupted between their tissue repair function and inflammation, can usually lead to tumor progression in cancer and monocytosis, or dysregulation of the number of monocytes or macrophages in the blood, for example. So understanding disease is one thing. Something that's slightly easier to do is to just understand biological function. So I've talked about phagocytosis. Turns out this is a nice example to which we can use a CRISPR screen to understand which genes are regulating that function. And this is work from Michael Basik's lab at Stanford, in which they had this very cute experiment in which they get those macrophage-like cells to basically eat up those magnets, those beads, that are then selected with magnets. And so because we have all of those perturbations, we can figure out which genes are enriched and entail a higher phagocytic activity or lower phagocytic activity. The kind of results look like this. So it's a genome-wide screen. Every point here is a gene, and we can tell whether there is more enrichment or less enrichment. And for example, genes that impair phagocytic activity or linked to actin filaments and this ARP complex that we know is involved in this process. So this is a very important point, and now we can understand again which genes are regulating one macrophage function. As I was describing, there is not a unique macrophage function. Macrophages can do many things. So the problem is if we can keep working with univariate measurements, the amount of biology we can understand or the impact on mechanism is usually limited. So we can measure the impact of genes on phagocytosis. We can measure the impact of genes on inflammation if we do an INO screen. We can, again, profile, for example, antigen presentation with another kind of assay. And we start building a multivariate map, but the problem is that we need to do one experiment for each readout. There is also this idea of limited interpretability. For example, there are many genes that impact phagocytosis, but we don't know how they do that. So we have limited insights on the mechanism of dysregulation. And we also have a resolution problem. For example, it could be that there is an emerging phenotype when we perturb certain genes that because we only care about one readout, we cannot understand. So technologies were invented to kind of go against those limitations. I'm going to be talking about two of those. The first one is Perturb-seq, in which we can take those pooled cells, this pool of cells with different genetic perturbations, and we can do single-cell RNA-seq profiling. Importantly, we can not only look at the gene expression in cells, but we can also see the abundance of the guides, which tells us which perturbation was in which cell. And this is one piece of technology. The other piece of technology revolves around imaging and optical pool screens, in which we can take this same pool of cells. So, I mean, it's not the same cells, but they come from the same pool of perturbation, for example, and we can place them under the microscope. And the microscope allows us for, of course, doing imaging. And here's similar story. Using chemical dyes, we can stain for organelles. Using antibodies and immunofluorescence, we can stain for specific proteins. That's what we call the image phenotyping step. But we also need to know which perturbation was in which cell. And this is actually technically very challenging to do in certain kind of cell types. But recently, we've been able to do that. And I know Gellert here, for example, worked a lot on that as well. And at Genentech, we also were involved in creating technologies for this. So it's usually done by in situ sequencing, and there are a lot of details I'm putting under the rug for talking about other things, but I'm happy to answer questions and point you towards literature. The way that you then would analyze those images, you need to segment and find the cells, and then calculate some features, for example. And based on that, you can see which features or abundance or like intensity of which channel, for example, is dysregulated upon different perturbations. And this is how we get so that's the other technology, optical pool screen, that allows at scale to characterize the effect of perturbations. And although both of those technologies are not better, but they provide more information than what I call a univariate screen, they still have their trade-offs. Okay, so optical pool screen, for example, is technically could be high throughput. You can just do more plates with a robot, and you can scale a really large library, and it's not that expensive. But having many colors is technically challenging, and you run into problems very quickly. Let's say tens of channels is possible, but already difficult. With Perturb-seq, you can have another problem in which you can do genome-wide Perturb-seq, but it's very expensive. So maybe you want to think about thousands of perturbations, but somehow with the advantage you have is that you measure many RNA species, right? The whole transcriptome. And so that's very appealing to characterizing the effect on gene programs, for example. And so what we were very excited to see is what's here. Okay, and so there are many things that could be there. There are not many things that exist. And what we thought about was pairing those modalities such that, for example, we can impute the expression of gene programs based on the imaging. And we'll talk a little bit about all of that. And so this is the point of this talk today, is to try to get the technology that would allow us to scale through perturbations, but also get high coverage, I guess. And we do that by putting those modalities together. And this is really, I want to highlight my collaborator Takakudo was able to develop the technology and make those experiments work. And also we've just been working hand in hand in this project, and it's been a really great collaboration. So now let's take a step back and let's see what we actually did. So Perturb-Pair is the name, I guess, of the framework, but it's also the name of the data set. And in this data set, what we profiled was we took those bone marrow-derived myeloid cells, we differentiated those cells, and we transduced them with an antivirus to get our pooled cells or perturbed cells. We then stimulate our cells with 9-hour lipopolysaccharide, which is a very established way of getting a response from those macrophages, stimulating their encounter with the bacteria. 9 hours is a good time point because you see transcriptional response, and you also see morphology response. Thinking about the time is a careful thing I'm happy to talk about later, but this is what worked here. And basically, we have two different libraries and two different experiments. We call that parallel experiment. In one experiment, we have a library of 1,000 genes that then we profile with site-seq, which is Perturb-seq with surface protein abundance as well. And we obtain a data set of 340,000 cells. And then with another library, which is bigger, it's not genome-wide, we had to downscale, but 3,000 genes and 8 million cells. And we have the images for those cells. I will detail a bit later. And then that tells us the impact on the morphology. Okay, so those are done in different cells, but they're linked by the perturbations. And if you have enough perturbations, it still tells you about how those readouts are related. And so this is the outline for the presentation, and the kind of things that we cared about when we even were planning this to work with this data set was, in general, how well do morphology and gene expression agree or disagree in their capacity to capture perturbation effects? Can we impute gene expression measurements, or at least up to a certain resolution, for perturbations that we only had the imaging for? And can we relate this to disease biology? So it's kind of a bonus, and we're going to touch on human genetics, hopefully. Yes, hopefully. And throughout, I'm going to try to tell some stories about how this technology is useful to recapitulate known macrophage biology. So let's start by talking about the Perturb-seq data. And here we'll take a little bit of a methodological detour because I've spent a lot of time thinking about how to analyze perturbation data, and specifically genomics data. So again, this is the experiment. So we take BMDMs, transduce them, and then activate them with LPS and profile them through site-seq. And we obtain gene expression, protein, and the abundance of guide RNAs throughout all of those cells. And let's say we already mapped it to the perturbation. So for every cell, we know what perturbation is in there. The important question again is what do we do with this data? And well, there is one thing that you would do is regression, okay, or taking basically pseudo-bulking, taking the average. So that's what was done in the first Perturb-seq data set with a bit of a nuance I will give, but essentially you want to know what's affected by which perturbation, and you can obtain after some processing matrices like this and this very nice heat map. So what you see here is a log4 change of expression of genes. So the genes here are the columns. The genes here are the row, the expressed genes, and the perturbed genes are the columns. And so what's really nice is that it's very straightforward to read, because you can look at one pattern of the heat map and you can say module 1, and those are JAK-STAT genes. So when you knock out those genes, you see a negative coefficient on the program number 1, which is antiviral response, which means somehow that those genes upregulate the antiviral response. And you can keep reading all those patterns and taking conclusions from them. One limitation is that to get to this, you actually need to clean the data quite a good amount. Like a lot of cells can be escapers, so like CRISPR has a certain efficiency. Some cells can also escape the perturbation. You also have some other artifacts. And so we would like to somehow instead think about not the pseudobulk, but really taking advantage of the single cell data for avoiding having to do a lot of manual, let's say, cleanup. And there are single cell methods. Some of them I've developed during my PhD, but even for those of you who better know PCA, for example, PCA is all meant about explaining the variance in the data with a few components. And the problem is that when we do this, we of course capture the dominant pattern, what I call here the dominant pattern in the data. And the problem is that what varies the most in the single cell data might just not be the perturbation effects. Okay, so we have kind of a mismatch between what those models are meant to do, which is maximize explained variance, and what the actual variance, total variance is in the data. So for example, this is here what you see when you apply SCVI to this perturb-seq data I was just mentioning. One huge part of the variance is cell cycle score. So this instance of problem has been documented in many perturb-seq papers. So here I'm just kind of like retelling the story. And it doesn't seem like it's a big deal. Okay, so why is it a problem? It's a problem because it makes the latent space less interpretable. If you were to cluster those cells, you would cluster them by cell cycle instead of clustering them per perturbation. When we think about recapitulating the function of genes through those perturb-seq screens, that's not what we want to do. Okay, so for example, here you have this cluster. In this cluster, there are several perturbations actually. So you have, let's say, cells exposed to many different perturbations. All those perturbations actually affect the M6A writer. But you can see that the cluster is polarized, if you want, and by phases of the cell cycle. And this is also true for this cluster down there. And the other problem that comes with that, besides interpretability, is specificity, meaning that the model uses a few latent factors to explain the variance in the data. And if we spend a lot of explanatory power to explain cell cycle, we don't use that to explain the effect of perturbation. So somehow we're not really maximizing what the models can do with the parameters that they have. And this is all happening because, again, those models have no knowledge of which perturbation was in which cell. But a regression model would know, because, and actually that's the only thing they would know, which perturbation was in which cell. So we have like very different type of like approaches here. One is regression, and one is those like PCA, or it's like single cell specific, or SCS, or variational inference approaches. And the idea is we would like to find a way instead to exploit this perturbation label inside of those single cell models to just explain the effect of perturbation, which is what we want. And so this is a dense slide, and the reason is that I just want to give you the gist of the ideas I've been thinking about these few years. But I've actually had a visit from Ethan when I was at Genentech, and Ethan was working on this very cool model called contrastive variational inference, in which basically you separate, you want to separate variance, and it's usually one of a lot of answers in this single cell data. Like, we actually need to think about what is the variance I care about, what is the variance I don't care about, and either you can regress it out or you can compartmentalize. In here, I think this framework is really nice because we can say we have the kind of variance that pertains to changes in gene expression in the control cells, and then we have another space in which we have the effect of the perturbations, and I've also taken those ideas and extended them because they used to be just a background data set and a target data set, but actually it's a bit more subtle. In the CRISPR screens, each perturbation is its own data set and induces different changes, and you want to know like structure across those changes. So I've done some work with Ethan, and all of this work, also Barbara Engelhardt was involved in doing like much earlier work before the CRISPR screens were that large. So this is somehow a lot of statisticians in single cell data really like that family of work. I'm not citing everybody here, but I've liked it also a lot as well. And I've applied it extensively, including to this data set here. This is what I would say for this slide. And if you want to have a look at those papers, please feel free to. You can also do a lot of theory, and I'll move on. So you can extend this type of work to multimodal data, and this is what I'm going to use here somehow. So again, we're going to have a background space in which it's a variation present across the control cells, a salient space in which is the variance induced by the perturbation. And this both latent spaces can be used to generate gene expression measurements, surface protein measurements that we have here, and with my collaborator in another completely different context in the T cells and chemical screens, we also use it for ATAC-seq, for example. And so what's really nice with those models is that it's very easy to extend them to including other modalities, etc. So here, I don't have time to walk you through what the background space looks like, what the salient space looks like. It's all really interesting, but let's focus on the salient space because we want to think about both modalities. So this is what the salient space looks like. And here are clustered perturbations according to that same space. And so you can see a lot of different clusters of cells, but they pertain to the perturbation. On the right, you see the pseudobulk. And so basically, we can find those clusters are very biologically meaningful, and this is already expected, I guess, for Perturb-seq screen. So we were very happy about that. And this is the analysis of the Perturb-seq data. In addition, we can also, of course, use those generative models to run differential expression. And when you have less cells, those models give you better power to resolve those effects. But essentially, you can still have those heat maps that people like to look at and that you can interpret. And so here are perturbation modules, and here are gene programs. So the gene programs are also very relevant for macrophage biology, and here to kind of do a back to the future or return to the past, I don't know which one. We have our JAK-STAT KOs that are down-regulating the function of interferon and antiviral program. You can look at many others. So now let's look at the perturb view. So our OPS is basically, again, we take those 9-hour LPS macrophages with a bigger library this time. And we have 24 channels, immunofluorescent channels, which is really, it's actually pretty large, and it's really involved to get to this scale. Interesting markers in there are, for example, P65, which is very important for NF-kappa B and macrophage activation, NOS2, IBA1, IFON alpha, and also SPP osteopontin, which is very important for certain aspects of macrophage biology in cancer, for example. And not only we need phenotyping, but we also need, if you want, genotyping. So we need to know which perturbation was in which cell. We do that with in situ sequencing. And we obtain, again, the statistics, so around 8 million cells with 1,000 features. So it's not just intensity, but you can look at correlations across features and do plenty of other engineered features like in Cell Profiler, for example. And we have 3,000 perturbations. So I'm just going to show you quickly what the data looks like. So our OPS is based on PerturvView that Taka developed independently. And you can see here, for example, this is one crop of the field of view. You can see many cells. And this is the first round of phenotyping. Then you need to do some destaining or bleaching and then restain again. And you keep doing this to get all the different channels. At some point, destaining doesn't work as well, or it's too aggressive. And so you got to be strategic about which protein you put first, etc. It's a bit involved. And then for the in situ sequencing, the way it works is by reading one nucleotide at a time and doing many cycles. And like this, you can read what is the sequence of the guide, and that tells you which perturbation was in which cell. So doing that, we validated our PerturvView by looking at imaging channels. And for each channel, we had the gene that that was responsible for this channel in the screen. So P65 KO or REL-A KO, of course, downregulate P65 imaging. And we can do that for all the other markers, and we had expected results. And so we can already find regulators of the proteins that we're staining for. And of course, with our features, we can do embeddings again. Okay. And so here there is a bit of an applied pipeline that I'm not talking about, but is not very remarkable. And you obtain some embedding that, again, the proximity of those genes in this embedding tells you whether perturbations are having same effect or similar effects, or same or different effects. And when you cluster that, you can again see some macrophage biology. And the point is that it's hard to tell how this is different from the transcriptome, for example, but in principle, those are things you would like to compare. So we're getting already into the theme, right? It seems natural to understand what is similar and what's different across those two modalities. And The one way to do this is, for example, by comparing the distance matrices. So here we've ordered the genes, the perturbation genes based on dendrogram. The dendrogram was computed based on the distance matrices on the joint, so by concatenating the both embeddings. And then on the lower corner here, you have the distance matrix on the transcriptomic space. It's a cosine similarity, actually, so higher is more similar. And on the upper triangular area, you have the similarity with the imaging. And so what you can see is that you can see the protein complexes in the middle, for example, and things seems pretty like coherent, and it's definitely more consistent than we were thinking about. So we're very happy to see this. What you can do is zoom in to see a little bit more. And so now you get this map that basically tells you, and you see the first thing you see up there is actually JAK-STAT, which is really nice. And so for all of those perturbations, you can tell how close they are in in the RNA-seq, how close they are in imaging. And then you can see which gene programs are changed upon these perturbations. And you can tell what is the change in intensity in the imaging of all of those proteins or a few selected proteins here. So it's hard basically to systematically, we don't have methods for actually systematically comparing. You can do correlation and it's better than random and it's statistically significant and qualitatively it looks like it's very similar, but we have a few anecdotes. So for example, here is a story where perturb-seq is better than OPS. If you look at these two clusters of perturbation that somehow cluster together, this is an example where the JAK-STAT members of the JAK-STAT pathway actually cluster with the components of the M6A writer based on the OPS, but they look very, very different in the perturb-seq. And yeah, I think it's pretty interesting because somehow those two pathways are involved, like JAK-STAT regulates the expression of those interferon-stimulated genes, and those genes get post-translationally changed by the M6A writer. But if you inhibit JAK-STAT, the gene expression is changed, but in both cases, you still have the same phenotype. And so the images look very, very similar, but the gene expression, you have expression of those ISG genes, which is very, very, very different. So that's like one type of situation for which perturb-seq, or like you have convergence on the imaging, but divergence in perturb-seq. And it's not always true that perturb-seq is systematically better. And that was actually very surprising for us. So here's what we looked at was out of our common libraries, how many genes are hit, like they have a strong effect or a significant effect in perturb-seq, but not in OPS, or in OPS, but not in perturb-seq. And so here, if you look at those perturb views, so OPS hit only, out of 1,000 perturbations, a third of them have an effect only in OPS, which to us sounded like, way too much, I guess. And you can look at this in many angles. You can look at the cell numbers. It's probably a combination of factors. But one factor that was a bit systematic was that you can look at the enrichments of those specific perturbations into the clusters, the modules you learn from OPS. And there are clusters that are super enriched and consistently depleted and not represented in the perturb-seq, even though they were profiled, they had no effect. And those, for example, cluster 3 and 5, where you lose more than 50% of those perturbations. And it's very interesting because in module 3, those perturbations are related to protein trafficking, and in module 5, they're related to mitochondrial protein and mTOR signaling. And when you look at which features are actually changed, for example, in module 3, all of those genes are actually upregulating osteopontin, SPP1, and SPP1 osteopontin is supposed to be secreted by the macrophages. But when you block protein trafficking, you might not be changing sensibly the expression if you want, but or like to a level that's not as remarkable, but you have so much accumulation of this osteopontin inside of the cell because it's not secreted anymore, because the protein trafficking machinery is broken, that somehow the images look substantially different and you have effect in OPS and not in perturb-seq. And for the module 5, what was interesting is that this is a phospho-specific antibody. And so basically there is difference in phosphorylation of that protein RPS6. Again, we didn't find a gene program that was significantly different across the two modalities. It could be that there is also power problem. So if you had like infinite amount of cells in the perturb-seq, maybe you would actually see genes that would be different expression. But at least with what we have, it looks like these are examples in which OPS has better power than perturb-seq. Okay, so the conclusion from this part. Overall, the similarity matrices or distance matrices are very concordant. There are some differences, but they're related to the biology of what you measure with the assay, so there is nothing somehow surprising here. There may be other factors that we don't understand. You can do an analysis of correlation across all the common perturbation to tell these gene programs are involved in translocation of P65, and there is a lot of analysis that I'm not showing here, but we have included in the manuscript. And overall, I'm just giving again a hint here of what's coming next. Because there is so much overlap, it seems likely that we can use PerturBio to predict PerturBSeq. So indeed, this is where we're going. So the plan is again to use morphological profiling as a way of like getting better measurements of gene expression. And so we're going to extend MoCaV very briefly to be able to make those measurements, and I would call that EB MoCaV. Empirical base, I'll explain in a bit. The two tasks that we want to do are very simple. The first one is we have morphology, but we have very low cell numbers, so we cannot confidently assess the effect of perturbations. We would like to somehow use the imaging as a prior to kind of denoise what's happening on the gene expression. And the other task is, oh, I do not have single cell gene expression at all, and I would like to impute it from the imaging. Okay. So for that, we basically, again, extended this MoCaV to be able to take into account the perturbation embeddings as well. So those can come from, I guess, any prior, but of course we use the OPS because we think it's a much better powered data to be able to real prior and to infer what's happening because it's done in the same system. And I will show you some results a bit later, but now I want to keep talking about this method. One thing I want to talk about is that this is not a method that will integrate the single cells of the imaging and the single cell of the sequencing. The reason we have done that, I just want to touch upon it, that it's very difficult. The variances are not exactly comparable, and it would require substantially more work. But for what we wanted to do here, which is a proof of concept, we can impute one modality from the other. That was enough to do this work here. So the way that the model and the inference mechanism work is as follows. So because it's a MoCaV model, we're going to need to somehow imagine what is our salient and our background latent variables. And so following the variational inference methodology, this is more for the technical people in the room. We kind of take as input our gene expression and protein measurement in the perturbation. We pass those through encoders and we can combine across modalities, and we obtain our estimates or our guess of what is the proposal distribution for the background latent variables and for the salient latent variables. And once we have a proposal, we can somehow combine this with a generative model. A generative model, just if you know where you are in the latent variables in the latent space, then you can generate the gene expression. And so here, for example, for the perturbed cells, you will combine both variables and pass them through those decoders. And for the control cells, this is one part of the methodology of contrastive analysis. You never use somehow the salient variables in the control cells, okay? Because the control variation is only in the background latent variables. So this machinery is just somehow specifying more in details what I've talked to you about in the previous slides. This is just the omics part, and this is a MoCaV model. And now we're adding an upper layer with this empirical prior. Empirical is because it means from data, and it's an empirical prior because we will specify what is the effect of perturbation, and we learn it from data, but combining the data from the imaging or embedding or image embeddings or pseudo-bulk embeddings, I guess, of the perturbations from the images. And we will try to line it up with what's going on in the omics data. Okay. And by doing that, so again, this is purely based on the OPS. And by doing that, we can take in not only, so we can basically denoise and we can also impute. So if we have a perturbation for which we have the embedding from the images, but we don't have single cell, we can pass it through this and resimulate what would have been a perturb-seq somehow sample. And so this is how this model works. We've applied it to predicting the transcriptome for all our perturbations for which we didn't have perturb-seq. Okay. And here I'm showing you, so again, you can do those imputations at different levels. You can do the latent variable imputation, so we know where things are in our salient space, the space I've showed you before. And then you can visualize it and you obtain this. And you can see that the imputations lie next to the data. I would have liked for things to be out of the data distribution, but I'm assuming that maybe there are some things that look either there is nothing surprising in our library, or sometimes you just cannot impute that well, right? And it's also part of part of what it is. But you can still have a look at this and try to understand whether those imputations make sense. And I'll leave the benchmarking for later. So we just want to know if this is already useful, and that's what we did in the data exploration phase. So we're going to zoom into that part. And that part is interesting because it contains, you can already see, for example, in LAMTOR, you have LAMTOR4 that was profiled in the Perturb-seq, but LAMTOR 1 and 3 were imputed and they all lie together. It's probably because they have the same imaging output, and they have the same output because they're all part of their LAMTOR subunits. So the imaging looked the same, and it predicted the single cell looks the same. That makes sense. Similarly, for this nice cluster here, where you have only one profile perturbation, CCDC22, and it lines up next to all of the commander complex. And actually, all of those genes are involved in the same complex, and they all physically interact with each other. So it's like kind of a sanity check, like it's nice to see that this is happening. But we thought this was a bit limiting because at the end of the day, what we wanted to do was, what about, can we find a complex somehow that was entirely not in our, that was only with imaging and not with zero sequencing, right? And so we didn't structure our data set for that because, when you prepare an experiment, you don't always know what you want to show. But after some digging, we found this SRP complex here where none of those proteins have been profiled in Perturb-seq, but we had, of course, imaging. And then we didn't know what those proteins do, so we used the model. So again, this is looking at clusters in the Ceylon space, but then you can regenerate gene expression data for them, and you can recalculate log4 changes, and you can ask what is different or which gene programs are very specific to those perturbations. And when you do this and you look at those genes, you basically get protein homeostasis and ER stress. And well, it really lines up with the role of those proteins, because actually those proteins are involved in the regulation of aberrant protein production. That if this whole system is not working, you basically get you get stress, and this is an important part in the protein homeostasis. Okay, so somehow the predicted genes line up with the role, at least overall. And to validate at least this specific part, we did run a Perturb-seq validation Perturb-seq screen, in which we included all those proteins, and we look at which genes were enriched. And for example, because we were targeted on this hypothesis, we can look at all the substrate of this wrap, and we can see that they're downregulated after SRP knockout. And then we can also see for heat shock proteins that are relevant for the stress pattern here, that they're upregulated. Okay, so somehow this is a loose claim that with imaging only, by lining it up onto our space, we're able to have a broad idea of what's happening. Okay, I'm not saying we can get biomarkers, but it's just a broad idea at least of what's going on to those perturbations. So now an important part is, okay, this is all a bit approximate, so how can we be thoughtful about evaluating our predictions and putting numbers onto what's happening here, okay? So there are two ways. One that everybody in the room who has read maybe a machine learning paper about this topic will think it's simple. I have a Perturb-seq screen. I take out some of those perturbations at random, for example, or maybe by clustering by pathway. I do my predictions, and that's my test set. The model never seen those, and I can evaluate. We've done that, but you can also do something a bit more powerful. You can, for example, think about it in terms of like, I'm the one designing those experiments, so I have done all my predictions, and I have a diverse set of predictions. I cluster those predictions, and I pick a representative perturbations for everything in those classes, and then I evaluate the predictions on the new data. So I have a model that only saw some data. Based on that, I profile perturbation, and then I do them in the wet lab, and I check the predictions there. And so again, we take this, we pick only the cluster that were far enough from the control. And then in each cluster, we pick 2 perturbations that were already measured and 2 perturbations that were imputed. We wanted to have perturbations in which things are measured because we wanted to understand if we had important batch effects, right? So batch effects are a problem whenever you run perturb-seq twice. You kind of want to check that you have similar results. Otherwise, you don't know what you're benchmarking against. And so it looks decent. So we were honestly, I have no idea how this was going to pan out. So this is nice. And because we get this result, well, now we can move to the evaluation. Okay. And so the main finding of those evaluation, and it's the same thing on holding out, is that most important for predicting the effect of perturbation is about what comes into the model, and it's not what the model is, which is, I think, something that by now people might know about. So the model that performs the best is EB-MoCaV because it uses OPS data. But it's not far behind the linear regression, okay, that would take as input somehow the PCA or like those really nice embeddings from OPS and predicting the log4 changes. And then trailing behind, you have all the other methods that do not know about the OPS data. Okay? And so the key message here is that somehow, of course, any perturbation data on the same system will tell you a lot more about your system than any off-the-shelf prior knowledge from the internet, right? But those numbers are good, but they don't tell you how far off are you, right? Because perhaps we could have gone the same SRP like predictions, perhaps we could have done something good anyway. And so I really wanted to make this figure in which these are only the test perturbations. I know my gene program, so I've ordered by the same gene programs you've been seeing in the other heat maps. I add the dendrogram, of course, ordering the perturbations here. And this is experimental data, I guess, my log4 changes. This is what EB-MoCaV predicted. And so, close your eyes, open them 3 times. Like you can find places where, I've been looking at this for such a long time. But there are places where it looks decent, but of course there are places where it's not the same. Okay. So, and it would be interesting to actually have a look and try to understand what are the pathways that are missed and for which reason, or what are perturbations for which we cannot predict well. And those might be the discordant cases that I've shown you before, right? Where things converge, but the gene expression is different. And I think that's okay. But if you compare to a baseline that didn't know about OPS, it looks like this. Okay. So what this shows is that the EB-MoCaV here baseline or like the method that we propose is somehow potentially useful, but the other methods are probably not useful for some of these kind of predictions. Okay? And this is the main message I wanted to say. So, okay. So the conclusion from this part. So okay, there is no, one more thing. I didn't talk about the denoising because I'm running out of time, but the EB-MoCaV, because it's in a nice Bayesian framework, can also allow you to denoise the effect of perturbation for low cell counts, which you cannot do with a simple regression. Okay, so either way, the method also has its own appeal. But we can impute and denoise perturb-seq data. We were able to interpret those imputations up to some extent, okay? And much more work could be done of that. But we did find a cluster that was only profiled through imaging and in which we were able to have a broad idea of what's going on. And OPS embeddings are much more useful than any other features available. I guess it kind of makes sense, but it's good to show it. And so this perturb pair setup makes sense. Okay, thinking about a cheaper modality that is a really good anchoring for scaling up the number of perturbation you will profile and still get molecular or like program type readout. And we did that, but with a follow-up screen, which was really nice. And again, I'm not showing this here, but the denoising helps us in for perturbation with low cell counts. Their neighbors were not what we found basically in a sentence is that the obtained perturbation neighbors were not very relevant. And once you denoise, you actually recover like meaningful numbers in terms of meaningful neighbors in terms of like protein complex proximity. Okay, and now this is kind of like the bonus part. Yes. Okay. When should I finish?
10 minutes left.
Okay. So we're gonna get to the bonus part. So the bonus part is how can those now we have a map. Okay, it's not genome-wide. It would have been great if it was genome-wide. It's not possible. So we have this map, and we have predicted the impact of perturbations on gene programs across let's say, thousands of perturbations. How can we make it useful? Okay. I mean, and it's useful in its own right, but how can we maybe make it more useful? And something that I've always been really interested in doing was like related those perturb-seq screen and trying to think about how to relate them to disease. And this is something that's very, very difficult to do. So when I and yeah, we're gonna get there. So basically, to discover genes that are regulating macrophage disease, there is either you can think about non-coding variation in its important part, but it's not what I'm going to talk about today. Today, I'm going to talk about using human genetics data and coding variation. So you can, in the UK Biobank, there is a whole exome sequencing for hundreds of thousands of individuals, and you have around 1 million loss of function coding variants. And you also have a lot of traits. So in particular, IBD and type 2 diabetes, monocyte counts, some of the diseases I've been mentioning about. And monocyte counts is the best scholar example. I mean, monocytosis is maybe an important disease. I don't know about it too much, but at least it's important because it's a continuous trait. It's a quantitative trait. And because of that, you can have this nice effect sizes plot. And so you know that, for example, a missense mutation in BRD2 will heavily impact statistically, the number of monocytes somebody may have in their blood. And if you look at the CEBPB, this is one of the main transcription factor that's necessary for like differentiation into macrophages. So it's these genes. Some of those actually make sense. And so we really wanted to find a way to integrate our perturb pair, including our imputations, with the loss of function burden test scores. And this was a framework that was invented by my colleague from the Preacher Lab, Minato Ota. And I want to highlight this because this is something that I think we didn't think was really possible. And he was able to make this work on K562 with a red blood cell trait and the genome-wide perturb-seq data set. And so we basically apply this method on our imputed data. So again, let me try to explain how it works. So we have the effect of perturbations onto gene programs. It's really important here to think that we're going to want to know which programs are important for the disease. Okay, so perturb-seq data tells us what are the effect of perturbation onto programs. The human genetics data tells us what is the effect of perturbations onto the trait. And somehow we want to define two quantities. The first one is the program burden effect, which means a program remains a bag of genes. So inside of this bag of genes, is there enrichment, positive or negative enrichment? Is it statistically significant for upregulating or downregulating the trait? And then we want to know another quantity, which is what we call what Minato calls the regulator burden correlation. And it's basically when I look for a given program at all of the perturbations, do I always have the same sign somehow between my perturbation upregulates the program and it also upregulates the trait, or it downregulates the program and downregulates the trait, or the signs consistent, positively correlated or negatively correlated. Okay. And now if the signs are positively correlated, you would believe that the program burden effect should be positive. That would make the program consistent with the genetic data. And if we were in the lower part, it would be the opposite. Okay, so the concordant zones are somehow those ones. And so it doesn't mean that the programs are terrible if you don't have non-concordant effect. It just means it's just nice if they do, and we can try to interpret them. Okay, and so what we found with those monocyte counts, for example, is the following plot in which the effects are mainly concordant, and not all programs are significant. And so you can try to pick as a priority the programs that are significant for both of those metrics, and you can try to understand what they do. Okay. And so after a bit of heavy reading and summarization, you can map all the programs and whether they have a positive or negative effect on the trait. And you can also find the regulators of those programs with perturb-seq. And what this basically tells you is like the potential parallel ways of affecting monocyte counts. And this is really nice because it allows you to understand what are the components or what are the pathways that are contributing to potentially monocytosis here. But again, let's just say as a scholar case, monocyte counts. And you can do this for disease. So for disease, it's a bit more complicated. The correlations actually don't work that well for reasons I'm happy to talk about offline. but you can still look at the enrichment, and we're very happy that our program showed differential enrichment by disease. And I'm going to zoom onto two specific diseases, type 2 diabetes and IBD. So both diseases we know are involved overinflammation, but so it's like, a subtle balance of like overinflammation and downregulating some other pathway that are, for example, alternate activation. But there is, for example, this really nice Th2 polarizing antigen presentation, so program 11A, that involved a crosstalk with T cells. So maybe somehow that could explain a different mechanism of the disease that doesn't really appear at all in IBD. And in IBD, what is negatively correlated with the log odds of disease seems to be the more classical alternate activation of those macrophages. Another thing that's interesting is that if you look at the key regulators, I tried to put a star by the regulators we found only because we imputed with EB-MOCAV. So somehow this is a use case where knowing more regulators is very helpful for mapping out the core regulator of like the disease. And a lot of those genes actually that I'm highlighting here, you can find support in animal studies that say that or in model systems that say that those genes are causal somehow for at least a model of the disease. Okay. And so as a summary, so we can find that the programs we find that are regulators seem to be mainly concordant with human genetics data. Beyond, we can also find disease-specific programs that seems to be interesting and maybe could open a way of like linking Pertube-seq data and human genetics data, even though this is a field that deserves a lot more attention and work. And this is just a very preliminary application of Mineto's work. I will say that the imputations are useful here because having more regulators is able to give us a better map. And finally, I want to say, so we introduced Pertube-seq, which is an experimental platform for combining imaging and sequencing in perturbations. We found strong concordance between the modalities and that even the differences are interpretable. We introduced this new method that can impute Pertube-seq measurements, and we validated it with a follow-up round of Pertube-seq. We were able to resolve the role of the SRP protein complex, which was known. It's not something we discovered, but we were able to make sense of it, even though we didn't have Pertube-seq data. And again, we were able to map it with disease. So, and thank you very much. This is the end of my talk. Let's take questions.
Really great talk. So question about, you have that overlap between what you obtain from Perturb-view versus Perturb-seq only. And yes, I think you mentioned that there's some power considerations, of course, but I'm curious about those 16 or so that you see only in Perturb-seq. And
yeah.
Is there, can you tie that back to the panel that you're using for imaging?
I think it was so few that I just looked quickly for like enrichment and it didn't look significant. So it might just be, there might be some randomness to the threshold you're using for significance of perturbation effects. Yeah, I think it was, the numbers were too few and I couldn't tell, but theoretically you should find things like this as well. But we didn't, 16 is very low. I think it's much easier to do this enrichment when you have 300. Yeah. But in different circumstances or with different maybe staining channels, and maybe that could also, yeah, I agree. If you were able to do that, you could maybe know how to change the panel, and yeah, that would be interesting. That's an interesting problem.
I have a related question. Did you evaluate how much the morphological features alone are driving the
Yes, so in this work, we have the actin filaments, like the cell painting channels, and we also have the proteins. And so I haven't. What's really interesting is that actually the energy distance correlates really, really well with across those two. So by masking the channel, which means that, and we haven't completely resolved this question, so I cannot answer your question rigorously, but my hunch is that it's just a cell painting has already a lot of information that there is an effect on the perturbations. But without the individual proteins, I don't think it's as easy to interpret what's going on. Yeah. Which was also surprising. But we haven't done the exact experiment of reassessing significance and checking back against that plot. That's why I can't say for sure. But looking at again how those energy distance by across 4 channels, imaging channel versus all of them, they correlate really, really well, except for a few proteins, and it's interesting. But that I think you don't really need all the other panels except for interpretability. Yeah.
So we don't need to do all the sequencing. Good questions.
Yeah.
Yeah, a very impressive follow-up question is, if I understand correctly, you said OPS is more accurate than sequencing, but I think these are two-dimensional data, so how you
Yeah. So I don't know what I said, but I really tried to say, imaging is better for certain things, and it depends what you image, and Pertube-seq is better for some other things. So we have I think the cases like this where you can better dissect the pathway with Pertube-seq than OPS is likely very true. It's true in many instances. I just showed you one of them. I think the map in OPS is much blurrier. I mean, you could probably see the embeddings. If you're used to looking at UMaps, which is not a good skill to have, it is blurrier. So in imaging, it is blurrier, definitely. So I don't want you to leave the talk thinking we should only do imaging. Okay, those two things are complementary. Yeah.
Yeah, I have a second question about the in situ sequencing. So, like in situ sequencing for the guide RNA identification, like I'm curious, like how what's the efficiency of that? Like you have
Yeah. So this is the piece of work that Gellert here and also Taka worked on, where it used to be on work only in cancer cells, and by using the T7 transcription, so in vitro transcription, they were able to make it work on basically any that I know, any primary cell types, for example. And so that's why it works in macrophages. Otherwise, we wouldn't be able to do the screen. After all the phenotyping, there wouldn't be any RNA left because the bleaching is very aggressive to the RNA. So those will be degraded, and then you would only get 20% or 10% of the cells. But somehow after the in vitro transcription with T7, and it's called the zombie protocol, you get a really nice recovery of like 90%. Yeah.
Sure.
Sorry. Yeah.
Oh, so
and then after you. Yeah, go ahead.
In the generative process in Vocabee, I noticed that you also included the reconstruction of the side seed. Is there any motivation or marginal help you found that adding this proteomics?
Yeah. The embeddings with the side seq look much better actually.
Okay.
Yeah, so I always say gene expression. I didn't do the extra work of knowing when Pertube-seq separates well or Pertube-side-seq. Is it because of the RNA or the protein? I haven't done that extra work yet, but here they're all collapsed together. In general, I can tell you from experience in other projects that there has been some cases where some defect of perturbation can be much better differentiated with the surface proteins. Depends what you're looking at. Yeah. So.
Thanks for the talk. I wanted, just out of curiosity, when you did your second Perturb-seq out of the data set that you must have left for the test set, did you see any perturbations that in the embedding were clearly distinct from everything that you've trained on? And how did the model do on those kind of perturbations that it had absolutely no training information?
So I guess the way that we generated this test set is we pick clusters in which there were always 2 measured things and 2 imputed things.
Right.
So it was always close to the training data.
I see. Okay, so we don't have any perturbations that in the embedding were.
So and that's what I kind of said. Expressed my disappointment, and I said, we didn't find perturbation that in the predicted space looked completely different from anything else. So if we had found those, we would have wanted to validate those, but it looked like it overlapped a lot. And it's probably the way that we build those libraries. Basically, we started looking at a lot of the genome-wide Pertube-seq screens, and we picked all of those genes, and then we kind of biased towards a lower full discovery rate to get a more precise library, and that more precise library is 1,000 genes. The less precise is 3,000. But if you want, maybe if the bigger library was genome-wide, maybe the model could have been able to say that's really weird. But because I think there is like a lot of it's the same context. I don't think there is a lot of covariate shift between those two libraries. And I'm assuming that the model could be wrong when, of course, there is no data to relate imaging to sequencing. That would make absolute sense. Yeah.
There are no more questions. Let's thank Romain again.